All right, so let's talk about the results. The results and conclusions for this X-ray vision lab. So far, hopefully you have done all the other parts that were necessary. You should have all the materials list and calculation, pre-lab calculations and the procedure, and a data table. Let me actually go back into my notebook and show you what I have. I was talking about this earlier in class, but just to reiterate in case, or if you missed it, there's a couple of important structural components for data tables. So data tables should always have the title for what the measurement is. So for example, mass of barium nitrate, and the units are always in parentheses after it. We do that so that way we don't have to put the units in the actual table. So the table itself should only have the data, like numbers or whatever you need. So you'll notice that's true for all these. You also need a descriptive title for every data table. The idea behind this title is it should be not quite a full sentence, but kind of close, like a sentence fragment that explains exactly what's going on in this data table so that someone could look at it and with the title and all of the headings have an idea of what you were doing. So I decided to go with collection of barium sulfate by filtration because that's kind of what we were doing in this data table. There's probably other better ways to put it, maybe talking about, I don't know, maybe collection of barium sulfate from precipitation reaction could also work. The fact that there's filter paper involved means there's filtration, so we may not need to say that. I don't know, there's lots of possibilities, but that is one of the possibilities. And at least looking at this, we now know what this means. So to finish this lab, one of the things we needed to do was to get the mass of our filter paper that has the barium sulfate in it, so we could figure that out. And I already did that, but I'll show you again how I did it. So just grab my balance here, turn it on, yay, pop this on there. And it looks like 30.22-ish, which is pretty much what I wrote there, 23, pretty similar. Okay, so it has definitely gained mass compared to just the filter paper by itself, which makes sense. So we now know how much barium nitrate we have, at least in terms of mass. So what we can now do is the results. For the results, you're also going to need this results table. When I was in college, I, for some reason, learned that you can do that, like you shouldn't have data and results in the same table necessarily. Doesn't mean you can't, but generally speaking, it's good to separate them. So that's why I've separated them. So we've got our mass of barium sulfate, which you can figure out by just subtracting out the mass of the filter paper, pretty easy. Our theoretical yield, we calculated it. It should be 1.3. That was the 1.30 grams. That's what we were going for. However, if your actual numbers of sodium sulfate and barium nitrate were not, were a little bit different, especially if they were a little bit lower, you might want to recalculate that to get a more precise number. So something to consider. But otherwise 1.3 is about right. Maybe a little bit less, maybe a little bit more. And then your percent yield is going to be the actual amount that you collected divided by the theoretical yield times 100%. That calculation, that formula is in your notes. Okay. The next section is the conclusion questions. We can write those in there. Let me just grab this for you real quick. This lab sheet doesn't have all of them, but yours will be updated. Basically, I'm going to have you all skip number 3, but number 1 said is what like human error-based mistakes did you, or in this case, really me, what did I make? This would be things like not doing any sort of human error where we didn't do the lab perfectly. Things like spills or I don't know, whatever else you can kind of think of. Anything that you saw that you would have done differently to make this maybe work a bit better. The lab design flaw is more about like, even if you do this lab absolutely perfectly, there's inherently, flaws in the design. So that would be things like, the biggest one I can think of for this lab would be like the filter paper itself. Like this filter paper is maybe not the highest quality filter paper in the world. And some of the barium sulfate is going to get through. There's not much you can do about it. There's a lot of other ideas you can think about. Tap water, that's an interesting idea. Maybe there's something interesting. I use tap water, so that's kind of interesting. Number three, you obviously were not doing this experiment yourself, so we only have my data set, so you can skip that. Number four, basically how successful were you in preventing, one of the big ideas behind this lab was to minimize waste. So how successful were you? And then finally, and then also why is it so important in the chemical industry to reduce waste? And then the last one is, it's not on this, this was an earlier version, but asking you to draw pictures. So I'm looking for something sort of like this to just give you an idea so you know at each step of the reaction what chemical species were present and were they dissolved in water or solid or what. So like, for example, this is the filtrate. So that's everything that went through. What actually is this? It's obviously water, but is there anything else in here? And if so, what is it in here? What would we be able to find if I let this dry out? What could we collect? Okay, so I'm going to stop there, and that's pretty much the whole lab. So we'll give that one a try, and we'll see how it goes. And there you have it.